Okay, so this video is going to go over the steps involved in purifying DNA from our unknown algae sample. This is what we're going to be doing this week. This is going to take the majority of the time of what we're going to be doing this week. After we've gotten our DNA from our algae, we have one last step this week, which is to set up the PCR experiments. More about that in another video, but that shouldn't take more than a few minutes to do. So this is the bulk of what we're doing today, or this week rather. What it involves is a series of centrifugations, filtrations, and biochemical manipulations to separate the DNA in the cell from all the other stuff that exists in the cell. We want nice, clean, pure DNA at the end of this process. So first, a couple definitions of words and techniques that I'm going to be talking about during the course of this video. You should already know this. When we're talking about centrifugation, after centrifugation, you're left with a pellet, which is the dense material that's compacted down at the bottom of the tube, and a supernatant, which is all the fluid on top of the pellet. Now sometimes, and possibly in this lab, the pellet will not be visible to the naked eye because we're dealing with extremely small quantities. So when we do these centrifugations, I will help you with figuring out if you have a pellet, where you have a pellet, and what to do with your pellet. In filtration, because we're going to carry out two different filtration steps, anything that passes through the filter is referred to as the flow-through. That's pretty straightforward. There are times when we're going to want the flow-through, and other times when we're going to want to throw the flow-through away. So it's very important that you pay attention to what step we're on and whether you want the stuff in the flow-through or you can throw it away. If there's any doubt at all, please ask me before you throw something away. Again, I'll be right there for you, and while we're doing these filtration steps, I'll let you know whether we're throwing away the flow-through or not. But it helps if you're on the same page. All right, so each group is going to get their unknown algae sample at the beginning, and the first step starts out kind of like it did with isolating chloroplasts. It's a physical grinding of those cells. These cells may have cell walls. We don't know what kind of algae it is. Some algae have cell walls. So it's important to do a physical grinding of the algae with what we call a micropestle, and you'll do that right in the tube that you get the algae sample in. But it's basically just a physical breaking down of possible cell walls. Once that's done, you're going to add 400 microliters of a buffer called AP1. We're using a kit that comes from a company called QIAGEN. And when they develop these kits, buffers get weird names based on difficult information to know. It's not clear why this is named AP1. That information is kind of lost to the development process of the kit. I can tell you what it does. This buffer is designed to solubilize all of the cell membranes, any remaining cell wall. Remember, we have to not only release the DNA from the nucleus and the nuclear envelope, which is a double membrane, but also the plasma membrane, so that DNA then is free-floating in solution and not confined inside a cell. So you put that 400 microliters of AP1 in, vortex it. Then we're going to incubate that at 65 degrees for 10 minutes, mixing every 2 to 3 minutes. Now, between the time you've watched this video and Thursday, in other words, the next 5 minutes, think about why 65 degrees. What is that temperature for? What's that going to do? After incubation for 10 minutes, we're going to add a buffer called P3, 130 microliters of buffer P3. This buffer is designed to denature proteins, keep the DNA in solution, but denature any proteins. Then we're going to incubate on ice for 5 minutes. Again, think about why ice for 5 minutes after all of this. Once that's done, we're going to spin it very, very fast, 14,000 RPM, and I've forgotten how many minutes we'll spin that for. That should result in a pellet and a supernatant. The pellet is going to be any kind of cellular debris, cell wall, some denatured proteins, cell membranes, bunch of stuff that we don't want. Our DNA should still be in solution, so it should remain in the supernatant. Now, I've drawn this like you're going to pour the supernatant. You're not. You're going to actually use a pipetter to gently draw off this supernatant without disturbing your pellet. This pellet should be visible. From that, you're going to then deposit that supernatant onto our first column, our first filter. This filter is called a Kaya Shredder filter. Remember, this kit is from a company called QIAGEN, and so they named this filter a Kaya Shredder filter. Its real function is to filter out debris that we don't want. Again, cell membrane, cell wall, isolated proteins, other intact organelles. All we want to pass through will be our DNA. Our DNA will pass through this filter. So in other words, we're going to ultimately want this flow through. So we spin that at 14,000 RPM for 2 minutes, and that additional gravity will force the fluid through this filter. And at the end, you should have a bunch of crap you don't want stuck in the filter. Our DNA should be in the flow through. So this one is a keeper. Then you're going to add 1.5 volumes of AW1 buffer. Now, what this means is, let's say you had 100 microliters of flow through. You would multiply that by 1.5 and add 150 microliters of AW1 buffer. So whatever volume of flow through you have, multiply that by 1.5, and that is the volume of AW1 buffer you want to add to the solution. So you add that to the flow through. You can get rid of this Kaya Shredder column. You won't need that anymore. Mix it well. And then the next step is kind of a repetitive step. Chances are you're going to have more than 650 microliters total volume after you've added the AW1 buffer. The next filter can only hold 650 microliters, so we have to do this in stages. You'll take the first 650 microliters and load it on the next column, which is called a DNA Easy Mini Prep column. This column is specially designed to bind to DNA. The function of this buffer is to get DNA to be highly charged so that it sticks well to this filter. When you load your first 650 mils on, you'll spin that at 8,000 RPM for a minute. That will drive through the flow through. Remember, this filter is designed to bind to our DNA, so our DNA should stick to the filter, and this flow through we don't want. You can toss that. So you take the filter out of the top of the tube, pour the flow through down the sink, put the filter back in the same tube, add your next 650 microliters on top of that filter, spin again, discard that flow through. Hopefully that will get the majority of the volume you had from this step, just doing this twice. So now you have your DNA bound to the filter, and the same tube, but it's now empty. You threw away, you discarded that flow through, and now we're going to wash it a couple times. So we're going to add 500 microliters of AW2 buffer. This is a washing buffer. This is designed to rinse away any excess salt that may have stuck to the filter, but leaving our DNA on the filter. So after that spin, you can discard that that flow through, because our DNA should still be stuck to this filter. And we may do this twice. I believe it says to in your lab manuals to do this process twice. The final step, you're going to take the filter out of the top of that tube. You can throw that all away. That filter gets put in a fresh, clean 1.5 microliter microfuge tube. And to that then, you'll add 50 microliters of what's called molecular biology grade water. This is an ultra pure form of distilled water that we use in molecular biology. That's different than I believe what it says in your lab manual. That's important difference, and I'll talk about that in lab. You let that 50 microliters sit on the filter for 5 minutes at room temperature. That allows the DNA to unbind from the filter and go back into solution in that water. It's not a very large volume of water, so that it won't flow through the filter in that 5 minutes. It'll just sit in that filter, solubilizing all your DNA. Then we take that after the 5 minutes and spin it again, and get rid of that filter. You don't need that anymore. And you're left with your purified DNA. Put that on ice. You're going to need it to set up your PCR reactions, which is going to be the next step.